Let me introduce Dr. Dana Zakalik, Professor of Oncology and Director of the Nancy and James Grossfield Cancer Genetics Center at Beaumont's OUWB Medical School, a Michigan alumna and UCSF-trained oncologist. Dr. Zakalik has spent nearly 2 decades building Beaumont's cancer genetics program. She is a respected leader in cancer genomics, teaching future oncologists and spearheading statewide initiatives in genomics and prevention. Her expert perspective has also made her a sought-after voice in the role of genetic testing in cancer care. And there we have it.
Good afternoon, everybody. Lovely to be here. Thank you, Jill. Thank you, Karam, for inviting me. I'm a little bit of a different speaker at this conference because I'm a medical oncologist. I deal with cancer, breast cancer predominantly, but I also am deeply embedded in clinical and research work in the field of genetics, particularly cancer genetics. I run a program at Corewell, which has really spearheaded a lot of the discovery in identifying who's at high risk for certain cancers or hematologic disorders. And along with that, we do see some patients with endocrine disorders and do germline testing. So with that background, this is going to be a little bit of a different talk. I'm not a surgeon, although I like surgeons. My brother's a surgeon. So in any case, I have no disclosures to disclose. So I'm going to go over a little bit of background and then take you through a sort of bird's eye view of what genetic testing looks like for patients with pituitary tumors. And we'll subdivide it into patients who present with simple isolated pituitary tumors, not part of a big syndrome, but 2 or more individuals within a family, and tell you about the genes related to that. And then there is another category, so-called syndromic pituitary adenomas, that are really part of a larger constellation of clinical findings as part of a syndrome that is genetic and inherited. And I'll say a few words about what genetic testing really looks like from the clinic and patient perspective. So by way of terminology, because I know my talk is a little bit different, there are certain basic terms that I just want to reiterate. Many of you have heard them. So we know what a gene is. We all have thousands upon thousands of genes. They essentially make proteins, which then regulate a number of functions in our bodies. Germline is hereditary. So when we talk about, and this talk is really about germline, genetics also happens in all of our bodies, genetic alterations that are acquired after birth that occur with aging or with growth of tumors, or just simply spontaneously or related to environmental factors. But germline is what causes certain familial syndromes. So that's what germline. Somatic is that other type of genetics. And that's really, and we do this a lot in oncology, where we sequence tumors to understand the molecular fingerprint or the characteristics of a tumor. In oncology, we target treatment, screening, prevention, and all of that. And that's somatic. And while there is overlap between the germline and somatic genome, really most of, when people ask me to give talks, I know that they most likely mean germline, but somatic is very important. That whole field of precision medicine is about identifying changes in genes, be it inherited or acquired, that we can take advantage of and target therapeutically. A mutation, by definition, is a pathogenic variant. So variants are variation within our genes, but you can have a benign variant, which doesn't mean anything. You can have an uncertain variant where we don't know, it's kind of in limbo, or you can have pathogenic or likely pathogenic, and that, by definition, is what a mutation is. And that's really the terminology that I use in a clinic. I don't really call these mutations, but in layperson's terms, anything that is disease-causing is called a mutation. So in terms of pituitary tumors, you've already heard a lot about them. These are the most common intracranial neoplasms with prevalence listed. They account for about maybe 1 in 8 intracranial tumors. They can consist predominantly of adenomas that can secrete prolactin. We're not talking about those today, or growth hormone, and that's the second most common type of pituitary tumor. There's increasingly greater and greater understanding of the genetic predisposition, and that really parallels what's happening in medicine. We're doing more genetic testing for different types of predisposition. Certainly the pediatricians have been doing it for a long time for pediatric anomalies, prenatal genetic testing. Cancer is huge in terms of volume. We're testing, for example, every ovarian cancer, every pancreas, every advanced prostate, most breast cancers, particularly triple negative or young onset under 60, that's considered now young. Because there's just more benefit from understanding our germline genetics. In the field of endocrine and pituitary genetics, that is still a small part of what we do. And that is where the area of research really needs to continue to grow. But for now, what we understand is that genetic hereditary predisposition is still relatively rare when it comes to pituitary tumors, accounting for about 5% or 1 in 20 pituitary tumors. But it's important to take a family history to recognize who needs germline testing. That is still, unfortunately, not routinely being done by most of us in the clinic. We're too busy. Family history doesn't seem as critical for us to ascertain. And that's why I really always try to remind people, take a family history, look at the age of your patient, et cetera. So this is a summary. So we subdivide pituitary adenoma genetics into that, on your right, isolated familial forms, of which there is a well-known gene called AIP that I'll talk about. The second one in that burgundy column is much less common, or syndromic, where it's part of a known hereditary predisposition, like multiple endocrine neoplasia type 1, for example, which we love to ask medical students questions about because it's not talked about very much, but that is pituitary tumors are very important component of MEN1, but then several others that I'll touch upon. And so today you're going to get kind of a bird's eye view, if you will, of the genes that can cause predisposition to pituitary tumors. And this pie graph, and if I may actually direct you to what to look at. So again, the distinction between genetics in pituitary tumors that's acquired or somatic, and that's the orange pie graph, part of the pie. The gray, that 5%, is what is germline. So this little gray right here is what we're talking about. So that is the hereditary. We're not, don't be scared, I won't be talking about all these genes. We'll just touch upon the main ones. All of this in blue here is what is essentially not genetic or not believed to be genetic. So we know most cancers, thank goodness, are not genetic, be it acquired or hereditary. So that's that blue part of the pie. And of course, that's where a lot of future research and ongoing research takes place. Again, the orange is the somatic. Again, 40% of genetic alterations are going to be somatic. About 5% will be germline. That's what we want to identify because that's what can impact our sisters, brothers, parents, cousins, and those are the people who can undergo genetic testing and then be forewarned that they have a higher predisposition, and also to better understand the natural history of the type of pituitary adenoma in terms of aggressiveness, resistance to therapies, medical therapies, where surgery may be more important, et cetera. So this is just starting to also impact how we manage these patients. So I am going to skip this slide in the interest of brevity, but this is again separating between germline and somatic and also categorizing, but I think this is just to drive the point. So again, the genetics is either syndromic on the right, that's MEN1, but we'll talk about a few of the others like MEN4 that many physicians have not really heard about, but important gene that's been discovered that causes that, as well as several others. Again, I'll just touch upon these just to give you a flavor. And this is that so-called isolated familial, where you have 2 or more family members with a pituitary tumor, and there is a gene for that. They're not part of a genetic syndrome. It's just isolated pituitary, as the name implies, FIPA, familial isolated pituitary adenoma, and the gene for that is known. Okay, so let's talk about that. So familial isolated pituitary adenoma condition is the most common cause of familial acromegaly. Again, familial meaning 2 or more individuals. There are over 500 families described. These are all relatively rare syndromes. Accounts for about 2 to 5% of all pituitary adenomas. The definition is having 2 or more cases of pituitary adenomas in one family in the absence of syndromic features. And there are 3 potential causes of familial isolated pituitary adenoma, or is it FIPA or FIPA? anyhow, okay. So it's FIPA. Either you have a gene called the AAIP gene, and that is the one I'm going to talk to you about. That accounts for up to a third of these isolated pituitary adenoma families. There is a second gene here that is listed that causes X-linked acrogigantism, which can be germline or somatic. Or in majority of cases, you have a clinical definition. You've got 2 or more people with pituitary adenoma. You do germline genetic testing, they are negative, and those are still familial cases because the definition of familial is 2 or more people in one family, but the genetics is negative. Either it's a gene we haven't identified yet or some other epigenetic alteration or maybe metabolomics, or it's not necessarily all in the genetics. These are characterized, this syndrome is characterized by younger age of onset, often macroadenomas, often more likely to be growth hormone secreting. And this is the most prevalent identifiable cause of a familial isolated pituitary adenoma within a family. In other words, if we're going to do genetic testing and find a gene, this is the one we're more likely to find. That second one that I mentioned to you is less common. This is, the gene for this is known. It's a aryl hydrocarbon receptor interacting protein, very complex molecular biology that's beyond the scope of this talk. It is, like many of these genes, a tumor suppressor. So we all have it. It suppresses tumors. When there's a pathogenic variant or mutation, it can suppress tumors. And that's the general principle of most cancer genetics, is that they are tumor suppressors rather than oncogenes. So the pathology is you have a mutation, a change in sequence, it's pathogenic, it suppresses the normal tumor suppressing function, and you get predisposition to having a disease. Again, most of them are growth hormone secreting, younger age of onset, 2nd and 3rd decade, and the gene is well described, and it does exist on panels. When we do in the clinic multi-gene panels, this is for pituitary disease, this gene is on them. One characteristic feature is that people who have this germline mutation tend to develop more aggressive adenomas that are more resistant to medical therapy. Genetic testing is indicated for patients who meet the family history criteria of 2 or more individuals or young onset of developing a pituitary adenoma. And people can test as young as in the pediatric population because there have been reports of this gene being identified as young as age 4. In terms of surveillance, it essentially starts with clinical assessment, laboratory studies that are listed anywhere from age 4 to 30, including a pituitary MRI that can start baseline at age 10, repeat every 5 years until age 30. These are, by the way, consensus guidelines from professional societies. After age 30, it's less likely, so repeat MRI surveillance only if clinical or biochemical abnormalities. So it seems like you age out of the requirement for pituitary MRI, and then you do clinical monitoring for some of the sequela of growth hormone excess, and sorry for that typo. In terms of now switching to a syndromic example, by contrast, MEN1 is a hereditary predisposition to forming benign and malignant growths in different endocrine glands. And the mnemonic in medicine, medical school, is the 3 Ps. Pituitary adenomas are by far the most common, parathyroid hyperplasia, and pancreatic islet cell tumors. So not the typical pancreas cancer, but the ones that, the type of cancers that form hormones like glucagon and others. It's a rare condition with a prevalence here listed here. Prolactinomas are more common. The second most common are growth hormone secreting pituitary lesions. Again, this is the characteristic. These are benign, usually benign, sometimes malignant tumors. There are other manifestation in the adrenal gland with pheochromocytomas, non-endocrine organs, skin manifestations, et cetera. I'll show you a couple of pictures. And this is very different from most of the hereditary cancer syndromes that I test for, like BRCA or Lynch syndrome, in that most growth occurs in hormone-secreting glands by definition. This is a little bit about the genetics. Autosomal dominant For those of you who don't live in the genetics universe, that means that if you inherit one copy from one parent, you have the condition, but you may not have the cancer or the tumor, meaning that unlike recessive means you need one from each parent. So many people are carriers of recessive conditions and will never have an issue. But this is truly all you need to do is inherit one from one parent, and that's why we do test children. And we find a mutation in a gene called MEN1 in about 2/3 to 3/4 of people who meet clinical criteria for MEN1. So I have a family coming in next week that is negative genetically for this gene, but they meet the criteria based on those 3 Ps. If you have 2 out of the 3 Ps, meaning, pituitary, parathyroid, or pancreatic islet cell, then you meet clinical criteria and you are surveyed as if you have the syndrome. So genetic testing has always been somewhat of a debate because you can have clinical conditions that essentially mean you have the syndrome. Okay, so then there are other things beyond those 3 Ps, just for folks in the audience who may read about this. You can develop these gastrinomas. So these are tumors that secrete gastrin, which then leads to acid hypersecretion, and you get these intractable ulcers. If you have a young child with ulcer disease, think of this syndrome because it really is a condition where people oversecrete gastrin, which leads to hyperacid secretion, carcinoid tumors, thyroid tumors, adrenal, etc. And then these skin manifestations, including angiofibroma. So this thing on the right is an angiofibroma, lipomas, collagenomas. So there is a characteristic skin presentation. In the pituitary, you see adenomas in up to 60% of patients with this condition. They tend to develop in the 2nd to 4th decade. Usually they're not malignant. Again, prolactinomas are more common than growth hormone secreting tumors, but you definitely can see that as well. So very important with MEN1 to be thinking pituitary. So now MEN4, and I'm not going to go into nearly the detail because it's much less common, maybe 1 in several 100,000, but MEN4 is essentially similar to MEN1, but you're negative for the MEN1 gene. However, you carry this gene called CDKN1B, which is on the panels for pituitary tumors. Most people don't really read much about this relatively new gene, but you can see pituitary tumors in up to 37% of MEN4 cases, as well as other clinical manifestations, incomplete penetrance, which is true for most of these genetic syndromes, and any patient that looks like they're MEN1 but is negative for the gene, consider testing for CDKN1B. Carney complex, another rare genetic syndrome that causes pituitary adenomas. The gene is PRKAR1A. And the very characteristic thing about Carney syndrome is spot, and I imagine these are board questions in the ENT world, I don't know, they weren't on my boards, but spotty pigmentation, so big cutaneous manifestation of Carney complex, as well as these benign cardiac tumors called cardiac myxomas, acromegaly, thyroid tumors. You actually, being a breast oncologist, I perked up when I saw this, breast ductal adenomas. So next time my pathologist hands me a breast cancer specimen, I see this, I will perhaps think of looking at possible pituitary lesions, and then schwannomas. So essentially, any patient that presents with this constellation of unique, rather rare symptomatology may be tested for this gene. And this is embedded in panels for pituitary, so we don't have to be that clinically suspicious because this is a gene on many large panels. This is I want to mention paraganglioma syndrome. I actually see this in my clinic in genetics. So this is a hereditary predisposition to paragangliomas and pheochromocytoma, so HPGL, hereditary paraganglioma syndrome. If you then have on top of that pituitary adenomas, it's called 3PA syndrome. So essentially it's the combination of the paragangliomas and pituitary adenomas. There's a series of genes that cause this. They're called succinyl dehydrogenase genes. There's SGHA, B is bad, D, C, and then this gene called MAX. And again, without overwhelming the details, just need to know that there is this other set of genes that will be on the genetic testing panels. And again, these patients do develop pituitary tumors, but the penetrance is very low. So I would not be necessarily surveying all these patients specifically, but when you have this syndrome, HPGL, you're getting MRIs every couple years through the base of skull and the spine. So basically, you just include the pituitary fossa if you're suspicious that a patient may have this based on family history or clinical presentation. Dicer, we're almost done. Dicer syndrome, again, I see this in my clinic. I had a young child present with a cystic nephroma, which is a benign kidney lesion, and was positive for this Dicer gene. So we did a molecular tumor board on this case. So Dicer, the hallmark, is pleuropulmonary, sorry for the spelling, pleuropulmonary blastomas. So that's the most, and by the way, those are aggressive, lethal, pleural-based tumors that little kiddies can develop. We had a young child that had to be airlifted to Michigan for a pleural stripping procedure. So this syndrome is important. It's very rare, but it can cause pituitary tumors. So just beware that these pituitary blastomas can be in the Dicer syndrome, which again, rare syndrome, but if you are in a busy center, you potentially may see these cases. So the gene is called Dicer, and only do genetic testing if they manifest some of the constellation. Lynch is one of the things I see the most next to breast cancer. It's the most common hereditary predisposition to colon and uterine cancer. The genes are called mismatch repair genes. They repair DNA. They're called MMR genes. They're listed here. Pituitary tumors are a rare, albeit described in the literature, manifestation of Lynch syndrome. People say in the Lynch research world that Lynch can almost cause anything, and that's almost true. I see patients with sarcomas. I'm shocked that they're Lynch positive. So we don't typically screen, but I just wanted to at least let you know some of the more rare conditions that are reported in the literature as causing pituitary adenomas. Last couple slides, principles of genetic evaluation. What we do in terms of germline genetics is one has to do a thorough family personal history. That really is our starting point. We do these big pedigrees. We look for syndromic features so we know what genes to include in our panels. Look at family history if there's another pituitary adenoma, again, in 2 or more individuals. That, by definition, meets criteria for germline testing. We're testing all pediatric patients with growth hormone secreting tumors, young adults with clinical presentation, et cetera. Genetic risk evaluation, I want to say a couple things about it. Genetic counseling is really important. I have a team of genetic counselors who are amazing. They do these complex family histories. They explain to patients. We have to get legal informed consent. Germline genetic testing is bound by privacy laws as well as informed consent statute, which means we have to explain to patients. We have to be able to analyze the results. When you do these big panels, you sometimes get inexplicable or uncertain results. So we are responsible for making sure that patients get proper communication as well as their referring doctors of what is truly a positive, what is uncertain, what is benign, and how to manage these patients. I think this is an important part of access to care, and as we're doing more genetic testing, we're looking at novel delivery methods. We started our program with these hour-long family history ascertainment, and we can't afford to do that in terms of there aren't enough genetic counselors. So we now are using telehealth. We're using access to technology such as chatbots, which are not yet here universally, but they're coming. We need to keep a database. We need to enroll these patients into registries because these are rare disorders. So the only way we're going to help patients is really do research and make sure that we learn from what we see so we can help the next patient. And so this is the end of my talk. Summary, there's a lot more known about genetics of pituitary tumors. We're doing better at next generation sequencing. We're learning more about the genes. I gave you sort of a bird's eye view. And with that comes better management. If we understand that certain genes, the genotype-phenotype correlation, that certain genes lead to more aggressive phenotypes, which would potentially guide our treatment. And really in the field of breast cancer and Lynch cancer, we are treating these patients who have a molecular inherited predisposition syndrome with different therapies. So I know that's not quite yet universally being done in all aspects of germline genetics, but I think the hope of precision medicine is that this will allow us to do screening, early detection, understand and predict natural history, and then ultimately treat these cancers better, these tumors better. And there's hope for the future. So that is it. And I have my cell and my email address. So thank you very much.